Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, we are following chapter number 5, that is A Cellular Life. I am Ms. Nushaba from Army Burnham College for Girls. Before moving on to the next topic, first we must have a self-appraisal or assessment of the previous topics. For that purpose, you are supposed to answer these questions. What is the status of virus? How do viruses get their envelope? What is the function of lysozyme and where is it found in a bacteriophage? What is the role of protein spikes found on the surface of virus? What is meant by maturation in the phage life cycle? And what is the function of reverse transcriptase? If you are able to do so, then it's well, but if you don't, then this is a wake-up call. Be vigilant as you have to achieve very high aim. Life cycle of human immunodeficiency virus is our next topic. As you already know, this is the causative agent of AIDS. It was identified in 1984 by a research team from Pasteur Institute in France and National Institute of Health in USA. It was named HIV in 1986, and researchers agree that this virus came from the chimpanzees and gorillas in the African continent to the humans. Now, just to revise the structure of HIV, you can see this is the outer envelope, which is composed of a lipid bilayer along with spikes embedded in it, these green colored spikes which have a pinhead like GP120, and this GP120 or glycoprotein 120 is used for attachment to the body of the host, and the second part is GP41, which is required for the fusion process with the host cell membrane during the entry of HIV into the host cell. Inside you can see the purple colored capsid. Capsid encloses RNA genome as well as enzymes. The enzymes may include reverse transcriptase, which is used to convert the HIV RNA genome into DNA. The other enzymes include integrase and protease, etc. What is the primary host of HIV? The primary hosts of HIV are helper T cells, which is a kind of white blood cells which constitute your immunity. In addition to helper T cells, macrophages, monocytes, B cells, microglial brain cells and intestinal cells may also be affected by this virus, but all the hosts share a common feature, which is the presence of CD4 antigen. This CD4 stands for cluster of differentiation 4, and the name is given to a specific antigens, a specific protein which is found on the cell surface membrane of the host cells. So primarily it is the helper T cells or T lymphocytes. GP120, as I already told you, that it is required to bind specifically to CD4 receptor on host cell, whereas GP41 causes fusion of virus to cell membrane, and after fusion the virus particle is able to enter the cell. Its genome is exposed by the uncoating process. Now this diagram illustrates the life cycle of HIV. As you can see, there are many steps given. So we begin with the step 1, in which you can see this the RNA and reverse transcriptase enters the host cell by the uncoating process. So after the attachment, fusion has occurred, and then the introduction of the viral RNA as well as viral enzymes into the cytoplasm. In the next step, DNA copy of the viral RNA is synthesized under the action of reverse transcriptase, the enzyme which is found in HIV itself. So the function of reverse transcriptase is reverse transcription, because otherwise transcription means the synthesis of messenger RNA from DNA. But in the reverse transcription, the DNA is synthesized from RNA. So after this step, the viral RNA is degraded, whereas the newly formed complementary DNA, that is duplicated in the third stage. Duplicated means becomes double-stranded. Now after becoming double-stranded, now it enters into the nucleus of the host cell and it becomes a part of the host genome. The process is called integration, and the action is catalyzed by the enzyme integrase from HIV. After integration, viral RNA is now called as provirus. This provirus is now able to transcribe further RNAs, and these RNA molecules include new viral RNA genomes as well as messenger RNAs, which code for different proteins required for the synthesis of new HIV particles. So in the sixth step, you can see viral RNAs are translated, yielding viral enzymes and structured proteins. Some spike proteins are also synthesized on endoplasmic reticulum of the host cell, and when these are processed in the Golgi complex, they are transported to the cell surface membrane, and these will be used in the formation of new envelope. The envelope is basically derived from the host cell membrane, and this in the last stage, the assembly or maturation of the virus takes place, and in this last step, the viral particle is budded off. This viral particle is completely infectious, and it is now mature enough to cause infection in the fresh T lymphocyte cells or other host cells having CD4 receptors on their surface. This process of budding involves consumption of cell surface membrane of T lymphocytes. So the result would be gradual decrease in the size of T lymphocytes. So that results in the loss of function of these cells, and as you already know that T lymphocytes are very important components of our immune system. So the result would be the decrease in immunity, and this is the reason that the HIV patient, that HIV host organism that suffers from a syndrome which is known as AIDS. AIDS stands for Acquired Immunodeficiency Syndrome. Just to explain the life cycle, we can look at these steps again. Fusion of HIV cell to the host cell surface, HIV RNA reverse transcriptase, integrase, other viral proteins enter the host cell. Viral DNA is formed by reverse transcription. Viral DNA is transported across nucleus and integrates into host DNA. New viral RNA is used as genomic RNA and to make viral proteins. New viral RNA and proteins move to cell surface and a new immature HIV virus forms. The virus matures by protease, releasing individual HIV proteins. The next video would help you in understanding the life cycle of HIV.
The human immunodeficiency virus, or HIV, destroys cells of the immune system, making the body vulnerable to a host of diseases. HIV has surface proteins called GP120 that attach to cells that have CD4 receptors on their surfaces. CD4 is found on the immune system's helper T lymphocytes and on scavenger cells called macrophages. The binding to CD4 and other cell surface proteins called co-receptors, not shown here, allows the virus to fuse with the cell. The lipid membrane of the virus incorporates into the cell's membrane, while the viral core enters the host cell. During entry, the core uncoats, allowing the viral genetic material, which is RNA, to enter the cytoplasm. In addition to RNA, the virus carries enzymes that participate in the first few stages of viral replication. Before the virus can commandeer the cell to make more viruses, viral enzymes must first convert the viral RNA into DNA. The viral enzyme reverse transcriptase copies the RNA into complementary DNA or cDNA. Reverse transcriptase has a high error rate and frequently leaves mutations in the copied DNA. The mutations result in variant forms of HIV, which allow HIV to evolve quickly. The viral RNA is destroyed by another viral enzyme called ribonuclease H. Reverse transcriptase then synthesizes a second DNA strand, using the first strand as a template. The double-stranded viral DNA enters the nucleus with several types of viral proteins, including an enzyme called integrase. Integrase enzymes splice the viral DNA into the host cell's chromosomal DNA. The integrated viral DNA is known as a provirus. The synthesis of the viral genome begins with the transcription of the proviral DNA into RNA. The viral RNA contains the code to produce the capsid proteins and other proteins needed for viral assembly. In spliced form, the RNA also codes for the envelope proteins and other auxiliary proteins. Envelope proteins are produced in the rough endoplasmic reticulum, ER, and then move through the Golgi apparatus before arriving at the cell surface. Full-length viral RNA molecules, along with all the other viral components, assemble at the membrane and bud off from the host cell. Although it's not clear exactly how HIV curtails the immune response or causes destruction of helper T cells, the viral infection ultimately results in loss of virtually all functional helper T cells in the immune system.
So this is how individual gets AIDS after the attack of HIV. The symptoms of AIDS appear in 3 stages. Stage 1 is asymptomatic carrier, in which the common symptoms are fever, chills, aches, swollen lymph glands, and itchy rash. These symptoms disappear and there are no other symptoms for 9 months or longer, but the individual is highly infectious during this stage, and the standard HIV blood test for the presence of antibody becomes positive during this stage. The second stage is AIDS-related complex or ARC. The most common symptoms are swollen lymph glands in the neck and armpit region as well as groin region, but the other symptoms include night sweats, persistent cough, flu, diarrhea, loss of memory, depression, and inability to think clearly. The last or the final stage is full-blown AIDS. During this stage, there is severe weight loss and weakness due to persistent diarrhea, and usually one of several opportunistic diseases may attack the patient. Now, what are opportunistic diseases? Since HIV does not cause any disease nor kills any person, it only destroys T cells of immune system. So the decrease in human immune system is the opportunity which helps in attacking the other diseases, which help in attacking of the other diseases to the host patient, to the host organism. These opportunistic diseases may include Kaposi's sarcoma, which is a kind of skin cancer or skin lesions, fungal infections, viral infection, gastrointestinal diseases, respiratory diseases, eye diseases, nervous system diseases, etc. may attack. Now about the treatment of AIDS. Although the complete cure is not known, but the purpose or the aim of the HIV treatment is to reduce the viral load. Now, what is viral load? It is the quantity of virus at which it is detected in an organism. And the treatment depends upon the symptom status of patient, whether it is symptomatic carrier in AIDS related complex stage or in full blown AIDS stage. It also depends upon many other factors, like the decision. It also depends upon the status of patient, so the CD4 count, the viral load, and the wishes of the patient. A combination of medicines that is called ART, antiretroviral therapy, is used just to increase the lifespan. Control measures for AIDS are more important, as by preventing the transfer of body fluids from patients to unaffected persons must be done by avoiding the use of used syringes, proper screening of blood before transfusion, avoiding the sharing of toothbrushes, blades, and towels with anyone, the sterilization of surgical instruments before use. In addition to AIDS, many other viral diseases are also there. The most common ones are hepatitis, herpes, polio, cotton leaf curl disease. Hepatitis is basically the inflammation of liver, but it includes other symptoms also, and it is generally caused by viral infection or sometimes due to toxicity of drugs and certain other diseases. Certain other causes also may be there. It may be present in acute or chronic form. The difference between an acute and a chronic infection is that an acute infection is very recent or very sudden, and chronic infection is that which is persistent, old, and slowly progressing. The most common causes of viral hepatitis are 5 unrelated hepatotropic viruses. Hepatotropic viruses are those viruses which replicate in liver cells. So these are hepatitis A, B, C, D, and E. Hepatitis A virus is caused by HAV virus, and it is transmitted by fecal oral route. Symptoms include fever, anorexia, nausea, vomiting, and jaundice. Whereas treatment, no antiviral therapy is available, but prevention may be done through a vaccine which contains inactivated HIV. Observation of proper hygiene is also very important. Next is hepatitis B or serum hepatitis virus. Serum hepatitis disease. It is caused by HBV, that is hepatitis B virus. Symptoms are similar to hepatitis A. It is transmitted by 3 methods, that is via blood, sexual contact, or from mother to newborn. The treatment is alpha interferon, and prevention is done through vaccine and the proper screening of blood transfusions. Hepatitis C, caused by hepatitis C virus, kind of flavivirus. Transmission occurs via blood, and symptoms are just like hepatitis B. Treatment is a combination of alpha interferon and ribavirin. For prevention, there is no vaccine available, but blood transfusion must be screened. Hepatitis D is the only type of animal disease or human disease caused by a VIROID. What is a VIROID? VIROID is a simple infectious particle which is composed of just RNA without any protein code. It is transmitted via another co-host, that is HBV, hepatitis B virus, because it needs the presence of HBV for the entry into the liver cells. Symptoms are just like hepatitis B, but they are more severe, and treatment and prevention is also same as HBV. Hepatitis E is caused by HEV virus, which is a kind of just like HIV, and it is transmitted by the fecal oral route. Treatment and prevention is not available, and observation of proper gene would be important for the prevention. Next is the herpes. Herpes simplex is a superficial viral infection, and it is characterized by one or more painful fluid-filled sores or blisters. What is a blister? It is an elevation of skin, and it appears on skin or epithelium of outer openings of the body. And tingling, itching, and burning may be felt on the skin before the blisters appear, and you might have observed it in other people, as well as you might have suffered from this disease. Also, like fever blisters which appear around the edges of mouth, etc. These are due to herpes simplex virus. These sores can last from 7 to 10 days, and there are two primary types: oral herpes and genital herpes. Oral herpes is caused by herpes simplex virus type 1, whereas genital herpes is caused by HSV type 2. Oral herpes is transmitted primarily through oral secretions like saliva or physical contact with sores on the skin. It can also be spread by sharing objects like toothbrushes or eating utensils. Symptoms include the appearance of blisters on the lips or around the mouth, and treatment is antiviral drugs. Prevention is through avoiding contact with the affected area of the patient. Same is the case with genital herpes. In general, a person can only get HSV type 2 infection during sexual contact with a person who has genital HSV2 infection. So symptoms include the appearance of sores on the genital organs. Treatment includes antiviral drugs, and prevention is by avoiding contact with the affected areas of the patient. Poliomyelitis, the most common disease in the previous centuries, but is now controlled through the WHO program of vaccines. It is caused by poliovirus, which is an enterovirus. Enterovirus is that which affects through fecal oral route, means through contaminated water or food. Symptoms include it replicates in the oropharynx and intestinal tract and spread to blood and central nervous system, where virus replicates in the motor neurons in the spinal cord and results in the death of these cells. So, as a result of the destruction of motor neurons, paralysis occurs, and so this damage is permanent and it is not reversible. Treatment is not available. There is no antiviral therapy, but prevention may be done by using two types of vaccines, any of the two types. One is called killed sac vaccine, or also known as IPV, that is injectable polio vaccine, whereas the other one is called live attenuated or Sabin vaccine, or OPV. OPV stands for oral polio vaccine. So polio drops, basically, these are OPV. This SAC vaccine or Sabin vaccine are named after the scientists who discovered these types of vaccines for the first time. Next is the cotton leaf curl disease. Cotton leaf curl disease is a serious disease of cotton, and it is caused by cotton leaf curl disease complex viruses which are found in the Indian subcontinent, and these are bigomoviruses transmitted by a vector which is called whitefly, or the scientific name is Bemisia tabaci. Symptoms include the deep downward cupping of youngest leaves of the cotton plant. Treatment and prevention is done through the insecticide spray against insect vector. Now, what are prions and viroids? Students, these are new terms for you, but remember, these are also virus-like infectious agents, but they are quite different from viruses, and they are smaller and simpler than the viruses. These are infectious agents that do not use nucleic acid and proteins together, and Nobel Prize winner Stanley Prusiner discovered prions for the first time. Viroids are group of virus-like pathogens, much smaller and distinctly different from viruses. They consist solely of small single-stranded circular RNA molecules. They have no protein coat or envelope. Viral RNA does not code for a protein because they don't have a capsid, they don't have a covering. Replication of RNA involves a special mechanism of replication which is called rolling circle replication. Several plant diseases are caused by them, so they are mostly plant pathogens. For example, in potatoes, coconut, apples, etc. Hepatitis D is the only disease which is caused in man by a viroid. So this is how viroid replication occurs by a mechanism called rolling circle replication. The detailed discussion of replication would be done in the second year. But just to give you an idea how replication in rolling circle way takes place, you are here shown a video that would probably of some help for your understanding.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A a region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method. In this
Next is prions. Prions are so called because they are proteinaceous infectious particles smaller than viruses. They contain no nucleic acids, neither DNA nor RNA. Electron microscopy shows that these are filaments rather than virus particles. As you can see in this slide, normal prion and diseased prion. Normal prion is actually a normal functional protein in the host organism. But a misfolding of that protein would result in diseased prions. Now it would become highly infectious and may affect the nervous system, causing different neurodegenerative diseases. This diseased prion or normal prion, this was actually a host cell membrane, host cell protein. So it is coded by a single cellular gene of the host itself. So prions are basically result of some mutations in the normal protein codes, and so misfolding has resulted in the development of prions. Prions cause different diseases in man, like mad cow disease in cattle. This mad cow disease is also known as bovine spongiform encephalopathy because it causes pores in the outer covering of the brain in cattle and gradually results in the death of the appearance of mad seizures in the cattle. As you can see in this slide, it's a neurological disorder caused by an abnormal protein called a prion. It affects central nervous system and causes brain and nerve cells to die. Prions are passed to humans through consumption of that brain, the brain of an infected cattle, and it can trigger the human form of the disease. This is how it spreads from cows to humans. Person eats contaminated food. Prions are found primarily in brain or spinal cord tissue from infected animal. And after a person ingests infected meat, prions spread to the brain through the body's lymph nodes and immune system, where they can remain in dormant form for years. Disease attacks nervous system and the outer layer of brain, which are called meninges, that develop tiny holes, and so it looks spongy. That's why it is called as spongiform encephalopathy. And death may occur later on. Prevention is by the destruction of infected form animals. And you see, prions are very, very resistant to inactivation by ultraviolet light and heat than viruses. Very high temperature, more than 900 degree Fahrenheit, is required to destroy prions. What are regulations? So animal products containing brain or central nervous system tissue aren't used as livestock feed, and consumers must avoid the beef that contains parts of cow's main nervous system. SCRAPIE is another disease caused by prions, and that is found in sheep. Another name is given in your book, that is kuru in humans, but it was found in a special tribe in the Papua New Guinea, where the people had a special ritual when somebody dies in their family, that they ate the brains of their dead people. So if the brain was infected, then the disease was, or the prion was actually transmitted to the individuals. Now it is almost rare and it is not found because cannibalism is prohibited. Now, next we come to the exercise, but before doing exercise, I must say that the chapter ends, but it doesn't mean that you cannot ask any questions. You can ask your questions if you find any difficulties through your WhatsApp group to the respective subject teachers. I hope you have understood the topics well. Now move on to the exercise. We will discuss only MCQs, and the rest would be your homework or your assignment. Do it on your registers and then learn it by heart. MCQs. First, viruses are considered non-living because they cannot reproduce independently. Option C. Second, which of these are found in all viruses? That is option C. Proteins and nucleic acids are found in all viruses. 3. Which step in the lytic cycle follows attachment of virus and release of DNA into the cell? So for that purpose you would have to recall lytic cycle. So after attachment and release of DNA, disintegration of host DNA occurs because the virus actually takes hold of the host machinery. Number 4, which of these is a true statement? So out of these 4 statements, the third one, that is option C, is correct. Viruses use the host ribosomes for their own needs because they don't have their own ribosomes. Number 5, which part of an animal virus is not reproduced in multiple copies? And that you must know that it is envelope. Number 6, RNA retroviruses have a special enzyme that transcribe viral RNA to DNA, and that enzyme is called reverse transcriptase. Number 7, which of the following illness is caused by a retrovirus? And that you must know, that is AIDS. Number 8, HIV primarily infects helper T cells. Number 9, poliomyelitis affects motor neuron. Number 10, HIV attaches to CD4 protein. And number 11, hepatitis D is caused by viroids. So for short questions, you will have to do yourself after going through the chapter thoroughly, and you will be able to do that inshallah. But anyways, if you find any difficulty, do let us know through our WhatsApp groups. Thank you very much for your cooperation, for your attention, and with this we come to the end of this chapter. And for Eid holidays, Eid Mubarak to all of you. Stay blessed. Allah Hafiz.